So I am at the barn in Eagle Marsh, and today is a special day. So first of all, who am I with?
Hi there, my name is Cami Sutter, and I am the Executive Director of Little River Wetlands Project.
Okay. Now your shirt says BioBlitz, so I'm assuming that's what's happening. And this is not the first BioBlitz you've been involved in. So can you tell us the history of Eagle Marsh and BioBlitz, and also what the heck is BioBlitz?
That's a good question. What is a BioBlitz? It's a fun name to say for sure. So the BioBlitz is an annual scientific study that's put on by the Indiana Academy of Science. It's a scientific group that chooses one location in the state of Indiana per year to study, to do basically a count of all the different kinds of species present at that particular preserve. So Little River Wetlands Project was fortunate this year in 2025 to be chosen as the study site. And so Eagle Marsh is the study location for the 2025 BioBlitz. We last did one in 2014, so we try to do one every 10 years or so, just to see what is happening out on the marsh, what has been the impact of the restoration work that we're doing. I mean, our goal is that when we took this flooded cornfield 20 years ago and planted it with native plants, that we're not only providing food sources for insects and pollinating plants for birds and bees and butterflies, but also habitat for birds and salamanders and beetles and all kinds of things. So today we had about 120 scientists from around the state come in for the day, and they work in teams of areas of their specialties. We've got a beetles team, we have a spiders team, we have a herpetology team studying the turtles, we have fish, we have, even my favorite is the snail killing flies team.
Very specific.
Very, very specific. And so it's really fun to see the very granular knowledge that these scientists bring, and also the passion that they have when they come in with this, oh, look at this little beetle larva. It's very exciting for them just to see what we have out in the marsh. The hope is that the restoration work that we're doing is moving the needle and providing supporting habitat so that the populations of these plants and animals can increase. So
I'm assuming that in the future there will be a formal written report.
Yes, there's a formal process because this is a scientific study done through the Indiana Academy of Science. There is actually a format that all of the team leaders have to follow to submit their data with very specific requirements that they have to include in their written report. That will be submitted to the state by October, and then it kind of goes up through the channels, I think through the state of Indiana, some various agencies, and then in about a year, that data will trickle back down to us, so we'll actually get to the formal report about a year from now. So it takes some patience, because we're all chomping at the bit to hear what the outcome is, but really useful for us, though, to have these two formal reports.
Well, thank you, and congratulations for such an exciting event.
Yeah, thank you very much. Our pleasure and joy to do it.
Hi, I'm Catherine Gwynn. This is my first BioBlitz, but I'm a student at IU Bloomington. I'm doing their dual degree program in a master's in public affairs and a master's in environmental science with a concentration in water management, because my ultimate goal is to work towards improving Indiana's water quality, because I feel like I can see the direction we're heading, and I want to get us back on a path that will keep people safe. I'm very much enjoying my first BioBlitz. I've joined in with the group for snail killing flies, which I'd never heard of before I came here, but I've learned so much from our team lead, William Murphy, I think is the name, and it's just so exciting to learn from someone with so much experience. They're such fascinating organisms, and I'm so excited to learn more as the day goes on. We've only found 4 so far, likely because we need to find the ideal habitat. Because the impression that I've gotten is that they're a very marsh-dependent species. They they're looking for snails, and they're looking for water that isn't too deep for that, like they'll drown or they'll get eaten by fish. For snails, they're looking to not be eaten by fish. They're looking to lay their eggs on a snail so that their larvae can go in and eat the snail. There's like around 2, and I just learned about this today, part of the appeal of this event. They go in and they eat the snail. So they have to find the snail first of all. And they like to do that on these kind of short grasses, not really the non-native grasses, is the impression I'm getting. So I imagine part of the reason we're having trouble finding them, it has to do with the fluctuation in habitat that's been seen around here. I've learned recently that there was a drought just last year, and now there's a fair amount of water, but it seems to have, I get the impression that's changed locations, but that variation likely means that they've either moved to places that were different than we found them before, or they have had trouble moving. And that's kind of the plight with wetlands, is they're incredibly important habitat for so many species, and they're incredibly important to humans as well. But they're changing because the world is changing. So it's about understanding the way they work and how they can benefit both us and the natural world, and understanding the systems that are at play to make sure that we take care of things. Because wetlands act to both help with drought and with flooding, because they absorb water and they release it slowly. So the better our wetlands are doing, and thus the better our snail-killing flies are doing, the better it seems to be for all of us. So I'm just enjoying learning about all the different connections between systems I've learned about in school in the field. And it's been an incredible event so far. Thank you.
I know, like, lots of little electricity. Okay, respirator ready?
Oh, it's got an X there.
Here's an adult, tiny stuff out of here. And then we get the big stuff out of there and out of here. I think we'll be ready to go.
Are we splitting here now?
Down the creek, and we got some to germinate.
Was it from Indiana?
No,
Somewhere else?
just going north.
Okay. There's the kind of system.
Oh, yeah. Yep. Okay. Yeah, it was a trail, like a tree to
How are you? Right from the camera, the other way.
Study that we did in the last BioBlitz. There were some that were prioritized higher than others, so we didn't get to 4 of the zones, I believe. But for the most part, I think we covered a lot of area with the rest. So we found a couple of good species out there. We found some forest gentian, some pretty good quality trees out there that were decent age, which is a refreshing sight to see, of course, whenever you're in places like that. I think we've got some good data for the area as a whole. Yes, I think that about covers it.
Can you say your name again?
Paul Chandler, yes, botanist.
So anyways, we had a great day today. We were trying to beat our record. So in 2014, there were about 88 species that were observed on this property. And so far, we've come up with 85. But anyways, probably some of our best birds today was a black burning walter that we heard, and that's the species that actually breeds up in the boreal forests of Canada, primarily in the coniferous forests, and it's one that actually goes through our state. It's a beautiful bird, and most of the migrants have already moved on, but we were lucky to actually have one still on the property. We also had a merlin, and that's falcon, and those are few and far between, so it's always excellent to see one of those. And then we also had a brown creeper as well, and that's another species that we normally wouldn't expect to see over here.
So my name is Michael Pickler. I was the head of the reptile and amphibian group, the herpetology group, and I love working with this group. How's that? Can everybody hear me? All right. So it was absolutely love working with this group there, because we always get a lot of people participating, even though we don't necessarily find a whole lot of species. But we did have a really successful day today. We found, let's see, I'm trying to pull this off the top of my head, so please forgive me if I'm doing this here. So several species of turtle. We found painted turtles. We found snapping turtles. Those were not pleasant, trying to get out of the traps, just so you know. Let's see, we saw the Blanding's turtle, which was kind of my key for the day there. We did look at Blanding's turtle, so that was a fun one. We saw a spiny soft shell, and we got to look at a northern map turtle as well. So we really kind of, across all of the turtle species that we normally found on the property. Yes?
Slider.
Oh yeah. We had a couple people also took a look at a turtle. They're pretty confident that they saw a red-eared slider on the property as well, which is an interesting one because that's not one that you would normally find in this watershed too. It's a species that has been introduced to lots and lots of different areas. And it's native to Indiana, but not the northern portion of the state where we are. So it could have spread into the area from a natural population, or it had been introduced here. We're not entirely sure. I personally did not see it, so I can't 100% verify that it was a red-eared slider, but people seem pretty confident about it. Yes, sir?
Was there a turtle laying eggs somewhere?
We did. Apparently, there was a photograph of a painted turtle, Midland painted turtle, that was also nesting. That's good. I found several destroyed nests along the back of the parking lot here over by the water. If you look carefully, you can find destroyed painted turtle nests on the ground there. So the raccoons have been eating well. So yeah, a really, really great day for turtles. We also found 2 of the 3 species of snake that we know of on the property. We found eastern garter snake. We also found DeKay's brown snake. It was interesting, we didn't see any northern water snakes today. You saw a northern water snake, did you? 2, fantastic. Okay, so I think we swept all the major snakes that we had. So that was the extent of the reptiles that we found. When it comes to amphibians, our group, as well as some other info that came in from a few other people. Let's see, we found a grand total of, I want to say, 4 species of neurids, frogs and toads. So we found American toadlets, kind of hopping around. We found a tadpole of a spring keeper. And we also saw gray tree frogs and northern leopard frogs. We may still have an opportunity. I don't know the frog population here all that well, but I wouldn't be surprised given the time of year here that it might be possible to hear bull frogs and green frogs calling at this time.
For spiders, we found a good number. As far as species are concerned, I really have no idea. I have to get back to the lab and ID everything, of course, just like beetle people will, of course, and many others invertebrates. But as far as families, I'd say the most common family that we saw, wolf spiders. A lot of wolf spiders, a lot of Pyrrhota wolf spiders that like to be near the water. Also found some big tigrosa wolf spiders that maybe are about 1 inch long. We found a few of those. But we did find a lot of jumping spiders as well. We found some beautiful paraphytopus jumping spiders. We also found jumping spiders that are actually beetle team. Found a really cool, I don't know the common names, I'm terrible. Marpissa formosa. I don't know what it is. Really cool spider. Looks neat. We also found a good number of sheet weaving spiders. So Linopiids, the ones that build little horizontal sheet webs amongst the grass near the ground. We saw a lot of those. Orb weaving spiders. Sac spiders. We found those. We also found, let's see, I think we found one funnel weaving spider. And let's see, crab spiders. We found crab spiders that like to eat pollinators and other flies and things like that on flowers. We found Pherideids, which are cone-footed spiders. I can just name all the spiders up here. We found many different types of spiders. How many species? I have no idea. But we did find a good number, and we found some pretty big ones, and we'll know, of course, in a few months what we actually found.
What is your name again?
My name is Mark Mill from the University of Indianapolis.
Hi, I'm Blake Talbert. I was on the fungus team. I'm not the team leader. That was Jeff Stallman. He kind of snuck out before this. They're on their way back to 30 to dry out our specimens. But yeah, we had a good day. We found a lot of rust fungus. We had a lot of rust fungus specialists on our team, so I think we got 20 of those. Some mayapple rust. If you guys aren't familiar with rust, they're like plant, I guess, they grow on plants. You know, it's like a fungal, I don't know what you call it. It grows on a plant. But a lot of times they're orange. We saw some ones on mayapples and on maples, and we saw some on some daisy fleabane. So those are all pretty cool. I think we got 20 of those. Not so many of the macro fungi, they're like really big ones. We saw some cool mycenas and stuff. We saw a stinkhorn, so that really stunk, but you know, we're happy to have that. Wasn't happy to have to split it up for genetic sequencing. I had to, you know, touch a lot, but you know, they had, what else did we have in there? Jeff was looking at a lot of small little asco species on like sticks and stuff, these tiny little cups that, you know, I would never have seen if you didn't point them out. So that was really cool to learn about. Trying to think, we had, oh, someone, Suzanne pointed out a, what is it? The wrinkled peach mushroom, which is really nice. So that was a really nice find. I'm glad we got that pointed out. So, yep, good day for fungus.
Hi, I'm Chris Worth, Beetle Team co-leader, and our brief tally is 30 at least 30 families. Like Mark mentioned, for spiders, we will need to take these back to the lab, do some ID work, key them out, but briefly, just conferring amongst ourselves, we have at least 30 families collected today. The malaise, and we also ran some malaise traps. So we had about 2 weeks of these traps. If you've not seen a malaise trap, it's like an inside-out tent. It's a type of flight intercept trap, and that those 4 collected what looks like bug soup. And so that will be hundreds of specimens to look through. Really excited to see what we have there. How this compares to the 2014 BioBlitz. Beetle Team then found 21 families, so we're ahead of that. And that was 64 taxa, and I'm confident we're going to best that just based on this family estimate. So overall, a good day. Excited to see what we find this evening.
Hi, I'm Jeanette Jaskula with Team Lepidoptera. So that's butterflies and moths, but most of our group is mothers. And for butterflies, it was kind of slow, you know, it was cold and windy today, so we're kind of behind where the group was back in 2014 when the BioBlitz happened here. I think we had about 5 or 6 species or so. But for moths, we had a pretty good night last night, but the temps are on the kind of the cool side. But I thought I would mention some of the names of some of the neat moths, because many of them have interesting common names. So we found the Batman last night, the lost owlet, we found it. the small bird dropping moth, and the slowpoke. We also had 5 Abbott's sphinx moths. Most of the moths we find are pretty small, but the Abbott sphinx moth is larger. There are even ones larger than that, but it feeds on grape as the caterpillar, and there's a lot of grapevine all around here. So that was really neat to find 5 of those flying around, at our lights. So we don't know the total number yet that we have until we all get back and process our photos and put them up on iNaturalist. But we will be mothing again tonight, so if you're sticking around, the sunset's probably a little bit after 9 o'clock, and it's probably not dark until 9:30 or even closer to 10. But we will be out there. It's going to be kind of chilly, but we'll be here again. So if you're around, you're welcome to stop by and hang out at the moth sheet with us. Thank you.
And the beetles.
So having assistants in the field pretty much changed the way that my day that I thought was going to be one way, it turned into something far more educational. In 2014, I wrote that this was a paradise for snail-killing flies. You could walk along the pathways and collect in the short aquatic vegetation anywhere in this property. Today, from snail-killing flies, I think we got 4 species. I had nearly 20 the last time. Everything's grown up. So the snail-killing fly habitat has been replaced by reed canary grass and rice cutgrass. There's no hope of using a sweep net in this stuff probably after the middle of June, because your net immediately fills with a lot of seeds. You have to pluck each and every one individually out of the sweep net. So as far as how the habitat has changed for snail-killing flies, it's pretty much a disaster here. But what's good for one thing is bad for another, like snail-killing flies, but the habitat has vastly increased for other insects, whatever likes reed canary grass or the other name that I just came out. Yeah, rice cutgrass. Both of which were intentionally introduced to Indiana because they grow in wet areas where more nutritious grasses won't grow, and they were meant for cattle fodder. So there you have it. I mean, they're out there doing a wonderful job of holding the soil together, but as far as snail-killing flies, what we look for, imagine on a snail-killing fly, is rather short vegetation where there's mud which will expose and strand the snails so that the snail-killing flies can get to them. So what we ended up doing was general biology today, which was fantastic. I learned as much from these 2 young women as I could have in an entire day in a college situation. We found everything from scorpion flies to that Virginia Tanuka, if I heard it, flyover birds. Seek came in very handy. It even got down on some plants to telling us what kind of infestation it had. Not just an oak, but an oak with this kind of a bacterial spot or whatever. But anytime you're in a BioBlitz like this, you can't help but absorb knowledge from the people around you. So, like Dr. Islam said, thank you for putting this together. It's great to rub elbows with people who know more about other subjects than you'll ever know. You can't help but leave a situation like this knowing more than you ever thought you would. So thank you to my compatriots here today, and I hope you will all come back for the next BioBlitz.
I'm Carl Stride, and my team was singing insects. And I always have to give the disclaimer that this is barely the beginning of the season when these things become active. I can match Bill. We had 4 species this time, which is 1 more than in 2014, but only because I now know how to identify a nymph that I couldn't identify. 2 of the 4 are not mature yet, 2 are singing. And so, pretty much the same report I give every year. I used to lobby to move these things later in the season, but I've given it. But I should say that I had 3 people with me today, 2 young students who were very helpful because at age 74, I can no longer hear a lot of the higher pitched singing insects, and so it's very helpful. And also, new faculty member at Purdue, Brianna, and I'm going to call on you now because, as I understand it, the bees, not bee team, but bees team, had an emergency and could not be here. But Brianna is a Hymenoptera person, and she did collecting and observing, and would you mind coming up and sharing a general summary of some of the things that you found? So, passing the torch.
I'm Brenna, new faculty at Purdue, and I've studied bees and wasps, so I was out collecting whatever bees and wasps I could find. So we had 2 bumblebee species, a Xylocopa species, which is the carpenter bee. Let's see, one Ceratina, which is the small carpenter bee. Hylaeus, so the masked bee. You might know of them from Hawaii. 7 are endangered in Hawaii, so the species that we have here are okay, but I got 2 of those. I got a couple halictids, so the green sweat bees, so the metallic green ones. They're really cute, and they'll just land and lick up the sweat on your hand, and very adorable and very photogenic. And I think there was an Andrenid bee as well. And I think that was it.
So there's a lot of carp. There's some big carp, 30-inch carp that you get with the electrofishing. And there's also a lot of white suckers, a lot of spotted suckers, especially in the deep parts near the bend of the ditch. The last spot over by the road, there was only like microscopic, really small. There's only goldfish and carp. That's it. A whole bunch of them. So I don't know if that pond dried out recently and it just got refilled. Because there's also like only really small, like sunfish, real small. So that might be the reason. But the ditch is full of minnows, creek chub, big-eyed chub, redfin, shiner, common shiner, sand shiner. A lot of bluegill. Most of the sunfish are bluegill. And then some ponds, especially the one over past Trail 6, it's completely different. It's predominantly pumpkin seed. And some parts of the ditch, you wouldn't find bluegill. You'd only find green sunfish, or long-eared sunfish. So the greens prefer the rocky parts. The long-ears prefer the more deeper and vegetated parts. And the bluegill, they don't care. So yeah, it was a pretty productive day.